So you may or may not be aware there's a few mutations now of COVID-19, so the coronavirus, that are getting around the globe. You might have heard of the one from Britain or one from South Africa. And look, you can't blame British people or South African people for those. These are just mutations that we know have probably come from those countries. But essentially now there's a lot of discussion of whether the current batch of vaccines will actually work for these mutations. But will they work? Yes, and kind of no. But I guess probably the big question some of you are probably asking, or all of you, well, firstly, what is coronavirus? But how does it get mutated? And what's this mean for vaccines moving into the future? So I think now would be a good time to take a deeper look into coronavirus and mutations in viruses. Roll the intro. What's up, nerds? It's your bro Tiny Hustler back with some snappy science in your face. Now this week we're looking at coronavirus and its mutations. Now just so you know, and I want to get this correct, we actually have an engineered version of the coronavirus. This is a version that lives in a lab somewhere that's called, it's called coronavirus. But they've given it a mutation that's called the N501Y mutation because science loves really catchy names. Now what's interesting about this N501 mutation is that it's very similar to the ones that are found in the British and South African mutations of the coronavirus. The good news is though that potentially vaccines can still be effective against these. This doesn't mean, however, that vaccines will work for all mutations in the future. And therefore we can end up in situations where you potentially could have a pandemic, or have a pandemic, as we have right now. So coronaviruses are a type of virus that's actually a long single strand of what they call RNA. It's like genetic material. And it's made up of letters, like alphabet letters, all joined together. But there's about somewhere between 26 and 32,000 of these alphabet letters joined in this one single strand. If you want to check out any of my other videos on coronavirus, you can check the link here. So you've got this long strand of what they call RNA that's made up of 26 to 32,000 letters, but how does that actually get mutated? First, I'm going to introduce you to antigenic drift. So viruses generally replicate, they make copies of themselves, and that's how they go through, say, the human body. But sometimes when they're replicating, they often make little errors in their coding because you've got all these thousands of letters that they have to copy each time. A few of these may not make a big difference, but as more and more of these mistakes in translations occur and build up over time, it can actually change the surface proteins, the proteins on the outside of the virus, which are known as antigens. It's these antigens on the outside of the virus that our immune system actually uses to recognize viruses when they enter the body and to remember them for the next time they come around. So therefore, if you've got copying errors and translation errors that's changing these antigens on the outside, this now becomes a mutation that the body may not recognize quite as well. So small antigenic drifts, which is small, copying errors over and over again, which changes the surface of the protein, the antigens, mean that vaccines can probably still continue to work because vaccines tend to work across lots of different parts of the surface. So if a little bit of it's different and mutated, a vaccine will generally be covering enough, almost putting it like it's a blanket, that will protect the body from that virus. But if you have large antigenic drift, so lots of letters get jumbled up and there's lots of copying errors, this could change large areas of the surface proteins, and eventually it will get to a point where the vaccine will no longer function because it won't have enough regions that it remembers to attach to. Antigenic drift is actually the reason that the flu vaccine needs to get updated every year. Because there's so many copying errors and they happen so rapidly that by the time it comes around in 12 months, you might have a different version of the flu that the current vaccine can no longer attach to those surface proteins. The good thing about coronavirus, especially COVID-19, is they believe that it actually replicates slower than other viruses, so therefore the rate that it makes these copying errors is actually slower and takes a lot longer. In some cases they believe it replicates 4 times slower than, say, the flu, so therefore you're not going to see these mutations as rapidly as the flu, where we see them happening every 12 months with antigenic drift. Scientists believe one of the reasons that it's slower but it has less antigenic drift is because COVID-19 potentially proofreads copies that it makes. So you have all these letters in a row, it copies them, and then it goes through and checks that it's done correctly. Another way that viruses can mutate is called antigenic shift. So before we had drift, now we have shift. Think skidding your car around a corner versus changing the gear shifter, where they have a dramatic change in the virus's antigens and therefore vaccines will just almost instantly no longer work. I feel like I'm saying drift and shift a lot here, and they're all just blending in together. One example of this is when you have two similar but different viral strains infect a host cell, so another cell in the human body at the same time. Sometimes the genetic material from the two of them can almost combine and make a new similar virus. Coronavirus. Because those two genomes are very similar, all those letters can combine together somehow, and they will refer to this as reassortment. The downside of antigenic shift is it can actually create a whole new strain of virus that vaccines and the immune system can no longer recognize or work against, and now you have populations that are vulnerable to this new disease. These are the sorts of viruses that can lead to zero immunity in the community. Could be a rap song. Immunity in the community. Coronaviruses actually don't have the ability to reassort, where all the letters from two different viruses get jumbled together because of the way their long stretch of letters is built. But they still do have the potential for two of them to infect the same host cell and their genetic material to almost get mashed together. So it's less likely to occur than with other viruses for coronaviruses, but this would be a case where you would end up with a novel coronavirus. So to go back to the original question, in the short term, yes, the current batch of vaccines we have should work against these new mutations because things are evolving quite slowly with these mutations. But there is the potential longer term, if this virus, the COVID-19 carries on for a long time, that there could be so many different mutations or antigenic drift, for instance, that vaccines may no longer work. But for now, we're in good hands. Be sure to smash like if you got some value from this video, and if you want more snappy science in your life, be sure to click the subscribe button and especially the little bell next to it to be notified of new videos. You've just been scienced.